The cell membrane is a fundamental structure in biology. It acts as a barrier between the inside of the cell and the outside world. But how did we come to understand this invisible boundary? Our journey begins in the late 19th century with the pioneering work of Ernst Overton. In the 1890s, Overton was studying the permeability of plant cells. He was intrigued by how easily some substances could pass through the cell membrane while others were blocked. Overton designed an elegant experiment to investigate this phenomenon. He used plant root hairs as his model system. He immersed these root hairs in solutions containing different solutes. He carefully observed which solutes could penetrate the cells and which could not. Overton made a remarkable discovery. He found that lipid-soluble substances readily entered the cells. In contrast, water-soluble substances faced difficulty passing through. This observation led Overton to a groundbreaking conclusion. He proposed that the outer boundary of the cell membrane, was composed of a layer of lipids. This was a revolutionary idea. It laid the foundation for our understanding of the cell membrane's chemical nature. Overton's work provided the first clue to the puzzle of the cell membrane's structure. 2 decades later, 2 Dutch scientists, E. Gorter and F. Grendel, took Overton's findings a step further. They sought to determine the exact arrangement of lipids in the cell membrane. In 1925, they conducted an ingenious experiment using red blood cells. These cells were ideal for their study because they lack internal organelles that could complicate their measurements. Gorter and Grendel extracted the lipids from a known number of red blood cells. They then spread these lipids as a monolayer on a water surface. They measured the surface area occupied by the lipid monolayer. They compared this area to the calculated total surface area of the red blood cells. Surprisingly, they found that the lipid monolayer's area was twice the surface area of the red blood cells. This unexpected result led Gorter and Grendel to a profound conclusion. They proposed that the cell membrane was not a single layer of lipids, but rather a bilayer. They envisioned this lipid bilayer as a sandwich with 2 layers of lipids. The hydrophilic heads of the lipids face outward, interacting with the aqueous environment inside and outside the cell. The hydrophobic tails point inward, away from the water, forming the core of the membrane. Interestingly, Gorter and Grendel made a couple of errors in their calculations. They underestimated the surface area of the red blood cells and the amount of lipid present. However, these errors fortuitously cancelled each other out. This led them to the correct conclusion about the lipid bilayer structure of the cell membrane. The lipid bilayer model proposed by Gorter and Grendel was a major advancement. However, it still lacked a crucial element: proteins. In the 1930s, Hugh Davson and James Danielli recognized this limitation. They knew that proteins were an integral part of the cell membrane. They incorporated proteins into the lipid bilayer model. Davson and Danielli proposed a model where the lipid bilayer was sandwiched between 2 layers of proteins. In their model, the proteins coated both the inner and outer surfaces of the lipid bilayer. These protein layers, they suggested, provided structural support to the membrane. They also facilitated the transport of molecules across the membrane. While Davson and Danielli's model was a step in the right direction, it had limitations. Their model depicted the membrane as a static structure. In reality, the cell membrane is incredibly dynamic. It is constantly in motion, with its components moving and interacting with each other. The shortcomings of the previous models led to a paradigm shift in our understanding of the cell membrane. In 1972, S. Jonathan Singer and Garth L. Nicholson proposed the fluid-mosaic model. This model revolutionized our view of the cell membrane. Singer and Nicholson's model retained the basic lipid bilayer structure. However, they envisioned the membrane as a fluid mosaic. They proposed that proteins were not merely confined to the surface of the lipid bilayer. Instead, proteins were embedded within the lipid bilayer itself. Some proteins spanned the entire bilayer, acting as channels or transporters. Others were partially embedded, acting as receptors or enzymes. The fluid mosaic model emphasized the dynamic nature of the cell membrane. It suggested that the components of the membrane, both lipids and proteins, are in constant motion. They can move laterally within the plane of the membrane. This fluidity allows the cell membrane to adapt to changing conditions. It also enables cells to interact with their environment dynamically. The fluid-mosaic model remains the most widely accepted model of the cell membrane today. It has stood the test of time and continues to be refined as we learn more about this fascinating structure.